Hello, listeners. Yamina here. Welcome to this episode of the Dr. GPCR podcast. Before we jump into this episode, I'd like to share a few things with you. The December and last Dr. GPCR newsletter of 2020 is now available. If you haven't subscribed yet, please visit drgpcr.com/newsletter. Also, don't forget to share the newsletter with your colleagues. We cannot wait to bring you brand new Dr. GPCR newsletter editions in 2021. Second, we will be taking a short break of the Dr. GPCR podcast in January 2021 as our family will be welcoming a baby boy. But rest assured, we are already working on bringing you brand new episodes, starting sometimes in February 2021 with an amazing lineup of guests. Also, stay tuned for a major announcement regarding the podcast. We're also proud to announce that we are pursuing consulting opportunities in the GPCR field. For help with your R&D project, please visit drgpcr.com/consulting or reach out by email at hello@drgpcr.com. And last but not least, we will be closing this first season of the Dr. GPCR podcast with a series of interviews with phenomenal female scientists in the field. We hope you'll enjoy it. Hello, everyone. I'm Dr. Yamina Bershish, founder of Dr. GPCR, and welcome to another episode of the Dr. GPCR podcast. Today, I have the pleasure of talking to Dr. Annette Gilchrist. She is an associate professor at the Pharmaceutical Sciences at Midwestern University. Hi, Annette.
Hello.
It's so nice to have you on the podcast. How are you doing?
I'm doing great, even for a pandemic. Beautiful sunny day in Chicago.
Well, we'll take sunny days anytime. I'm glad that we were able to, put this together and talk to you. So, can you tell us a little bit more about your career trajectory? See, it's the summertime, so I'm having a hard time getting words out. And what do you do exactly as an associate professor?
Sure. So those are two totally different questions. If I forget one by the time we get to the end, just remind me. Trajectory. So I would say, as most scientists, I have an unusual trajectory. I don't think anybody, or a few of us, start out saying, I want to be a PhD. So I started with the idea that I was going to go to medical school. I tried. I'm originally from Oklahoma, and I applied at the University of Oklahoma, and they weren't interested in me. I guess my GPA wasn't high enough, for whatever reason. And so I decided to actually go and get a master's degree because I'd heard that if you had a good GPA with the master's that you could usually get in. So I said, fantastic. I went to the University of Connecticut, actually, to get my master's degree because at the time, Oklahoma didn't have like a fantastic genetics molecular biology program. So I went to UConn and I got a master's in biochemistry. And I, at the time, was doing research on the toxic effects of acetaminophen on neonatal animals. And when I finished my master's, I actually took a job at Pfizer. So I worked for somebody named Peter Hobart, and the work was the first time I think I was ever really interested in science with my whole being. I thought it was amazing and fascinating, and I couldn't get to work early enough, and I couldn't stay late enough, and I wanted to know the answer to every question that we were working on, which at the time was looking for drugs for osteoporosis. So I became interested in bone, was really fascinated by bone itself and how it forms, and actually was thrilled. I'd gotten into medical school, was going to start in the fall, but I loved my job so much at Pfizer, I decided that I didn't really want to spend the rest of my life treating cancer, which is what I had hoped to do. I decided that I'd rather look for a cure for cancer. And so literally 2 weeks before school started, decided to go get my PhD. So thankfully, I was accepted incredibly late into a program at University of Connecticut Health Center, and I ended up getting a PhD in immunology. So that was years ago now, decades ago. At the time, I studied as an undergraduate. As a graduate, I studied tyrosine phosphatases that were activated by FNet-Lu, which happens to be a GPCR, but that was certainly not anything that I planned on. I was looking more at the tyrosine phosphatase signaling aspect of it. But when I looked for a postdoc, I fell in love with Chicago, which is where I still am, some many years later. And I got a postdoc with Heidi Hamm, who's well known for her work in protein-coupled receptors, and came to Chicago and looked at receptor G-protein interactions. So since my postdoc started in like 94, I've been working on GPCRs ever since. So.
Wow. And what made you decide to do a master's in biochemistry and molecular biology?
I decided it was my penance. I really didn't love chemistry. It was not something that I found to be intuitive, which is really funny because I teach med chem now of cancer drugs. So I feel like, you know, if I have a subject that I'm not great at, the only way that I can get really good at it is to teach it, I think. And at the time, the way that I learned biochemistry was to master in it. So while it was definitely death by fire, I think in the first exam, it was definitely something that I put to use in every aspect of my sort of scientific career.
Wow. Especially that initially you went to do a master's because you quote unquote just wanted to get into med school.
Absolutely. So no intent of using it like long term.
Which that's the irony. So when were you first exposed to working on GPCRs? Was it in Heidi Hamm's lab?
So no, I was actually getting my PhD because I worked on FNet-Lu. So like I knew the FNet-Lu receptor was a GPCR, and so knew about chemotaxis through G-protein coupled receptors, that kind of thing. But it was definitely not the sort of focus of the research at all. And in fact, when I told the people on my committee that I was going to go and work for Heidi, they were all like, well, you know, G proteins have already got their Nobel Prize. They're not going to get it again. They didn't know that Pavelka and Lefkowitz would, in fact, get it.
Exactly.
But I was just, you know, like signaling, to me, signaling in a cell is pretty interesting. So whether it's through kinases or phosphatases driven by GPCRs or non-GPCRs, I think it's, you know, they're all related and they're all going to talk to each other in one way, shape, or another.
Of course, of course. And how did the opportunity to work at Pfizer come up? You know, oftentimes students or grad students who are in academia, in their mind, the only way to move forward in their career is to do a PhD, a postdoc, and then hopefully become a professor. But oftentimes we don't have that information or infrastructure out there to understand that with a PhD, you can do so many things, including working for in industry. So how did that happen?
You know, it was probably just like an ad on the wall at the university. I honestly don't remember. I mean, I remember interviewing because it was like an all-day affair, and I had never been on an interview that lasted 8 hours, and I just thought that was like, really? How much more can I tell you? Or you say the same story 8 times for all the people that you meet. Why are you interested? Blah, blah. But it was pretty eye-opening. So I feel really fortunate that I worked for Peter Hobart. He really insisted that I go to many of the talks that were provided there. I met like many people that I would otherwise not, because the money, of course, at Pharmaceutical get really amazing speakers that come in and talk and that do consulting. And so I felt incredibly fortunate that I had that experience later on. And certainly I think the way that you think when you work in industry is often very different, right? So um, how you solve a problem when you work in academia is not the same way that you solve the problem when you work in industry. And so that was pretty interesting to me. I was really glad. I've always had sort of both sides of the coin, with both industry and academia background.
Phenomenal. Yes, and you're right. The 8 hour or all full day interviews are still happening. That's still the case in industry. And sometimes you go through many selection processes over the phone. Well, now the 8 hour interviews, I think they're kind of shortened down to 4 hours, but on Zoom. Still, it's still a long interview process. And definitely having that experience of both sides of the coin is very important, because I think in some way both sides kind of complement each other if we're looking at, drugging GPCRs, or coming up with a medicine that will be useful in patients in some disease setting.
Yeah, I think so. One of the things that Peter really instilled was the idea that, the whole point of a pharmaceutical company, though, is that you will make money, right? I mean, it is not something that you're doing because you want to cure cancer. You're curing cancer because it will make a profit, right, for a company. And so with just that in mind, just as an overarching goal at a company, you don't have that in academia. So if I decide, right, I see something interesting and I want to follow that discovery, I can do that in academia without any problem at all. If I want to change my receptor, I change it. If I want to study a totally random, non-GPCR related thing, that's okay. I can still do that.
Yeah.
And you just don't have that freedom in the world of industry.
Oh, definitely, because as you said, at the end of the day, the whole point of sustaining the organization is making a profit, is making money. However, what I would like to add to that, I feel like not all companies, but some companies still allow you to kind of have that some percentage of your time working on something that might lead to a bigger project. But if leadership pulls the plug, they pull the plug, and
No,
that's it.
absolutely. Even Peter had, like, some certain percentage of his time that he could study a side project. Yeah, absolutely.
Yeah. And then so you did your postdoc in Heidi's lab, and then what next? What happened next?
So actually, Heidi and I started a company. So I ran that company for the better part of 10 years.
Wow.
And it ultimately moved up to Wisconsin, and I was in Wisconsin for 2 years. And then I had twins. And when the company closed, then I returned to Chicago and ultimately took a job in academia, where I still am 11 years later. So my kids are now 12. And so I'm still here and still doing work on GPCRs.
So basically, you do have that really experience on both sides of the coin, not only working at big pharma, working in academia, but also running a company. Was it a GPCR focused company? Can you tell us a little bit about it?
Sure. So it was a GPCR company. It actually had two different names with two different intents. So it started out as Q Biotech, CUE Biotech, where we were looking for small molecules that were allosteric modulators of GPCRs. And the way that we identified the allosteric modulators was to measure GPCR G protein interaction. So you could identify something that could slow the turnover of GTP turnover with G proteins, and that would provide you with an antagonist, in essence, of signaling.
Yeah.
So that's how it started, and we received several SBIR grants, and finally, ultimately decided to raise money through venture capitalists. And so when we did that, we changed the focus of the company to be a tools company, where we would actually provide, as part of the process to identify the allosteric modulators, you first identified peptides that mimic the C-terminus of the G proteins. And so we would provide those as tools to pharmaceutical companies, for example, so they could choose to do the screening themselves, or we could do it for them. And so that company, once it got VC funding was called Caden Biosciences, and that's when we moved up to Wisconsin. One of the venture investors was a Wisconsin-based venture program. So we moved up there and worked on the project for a couple years. But ultimately, as a tools company, it was too difficult to try and sell that process. It's a relatively onerous process to make those peptides work in a high throughput screen. And so at that point, when the company closed, literally, I just came back to Chicago and enjoyed myself for the better part of 18 months as my kids were young, and then went back to work in academia.
That's great. That's great. And it's something, I think it's something that needs to be tried out. If you have a good idea, if you have a good infrastructure, you can get capital, why not? Why not try it out?
I think that's true. It's interesting. I've actually started another company recently that has nothing to do with GPCRs, but is related to pharmacogenomics. And it's because, you find a problem and you think you have a solution, right? I mean, I think very much it's the idea that you've done something, and if you've struggled with it, you've tried it 100 times and you find a solution to that problem, you think, I think somebody else might want this same thing. And so the question is, can you sell it? Can you make a profit?
Yeah.
And I'm sure that my experience at Pfizer really sort of helped me think about how you think about a scientific question. How do you make a profit out of it? How do you put it to practice?
Yeah, so it definitely shaped your way of thinking about science, which is phenomenal. I think it's rare that as an academic scientist, someone who started out as an academic scientist, you get the chance or you get that exposure early on, and then you decide to just go at it and try it out, which is phenomenal. I hope it'll inspire a lot of our listeners. Getting back to GPCR, so you've worked in Heidi's lab, and then you worked at these companies, and then you're back in academia. Do you have a favorite receptor or receptor family that you're currently exploring?
So, it's pretty funny that I'm pretty receptor agnostic. So
Okay.
I like all GPCRs. So I personally currently work on a chemokine receptor called CCR1.
Okay.
And I work on the free fatty acid receptor FFA2. I am working on, with another colleague here at the university, I'm working on the muscarinic M2 and M3 receptors. So, but in the past, I worked on rhodopsin when I worked in Heidi's lab, and I worked on CAR1, which Paul Insel talked about in his episode with you. And so, I think a lot of GPCRs are really interesting. He talked about the proton receptor. I'm fascinated by that because much of what we looked at when I was at Pfizer and looked at bone had to do with low pH, that the cells get osteoclasts get turned on only at a low pH. And so you wonder if there's a proton receptor that's driving that phenomenon for differentiation, certainly for cancer. So I think there's a lot of, I personally love to read about the adhesion GPCRs. It's like a hobby in all my spare time that none of us have, but I still do it because they're huge, and I think that's amazing, So I have a lot of GPCRs that I read about, but I personally work on mostly CCR1, FFA2, and muscarinic M2 and 3.
Wow. Well, I think it's researcher dependent, the love for, one specific family or, having all of these interests. But definitely adhesion receptors are really cool and really interesting, and also hard to study. I think it's one of the reasons there is not as much information out there as you would want.
Yeah, definitely.
So in what context are you studying, for example, the chemokine receptor, CCR1 or CXCR1?
CCR1 is the one I
CCR1.
look at. So I look at it in the context of cancer, right? So I'm back to cancer, which is where I started, right? I had an interest in cancer from early on. And I look at it in two aspects. So I look at its role in multiple myeloma. And I look to see, because bone is clearly involved in multiple myeloma, the patients often get lesions in their bone, and present because they have broken bones, having no other symptoms, and it's because their osteoclasts and osteoblasts are no longer in balance. So they have too few osteoblasts and way too many osteoclasts, way too active. So CCR1's been implicated in osteoclast differentiation, and so I'm looking for inhibitors. So mostly what I do is screening. I'm looking at inhibitors for CCR1 that would turn off osteoclast differentiation and potentially enhance osteoblast differentiation. So you could keep that in better balance in those myeloma patients. And I'm also interested in CCR1 because many cancers home to the bone when you have metastasis. So metastasis is actually what usually kills a patient who has cancer. It's like 90% of patients actually die from metastasis, not the original cancer itself. So prostate cancer, breast cancer, several cancers actually preferentially home to bone. And so I want to know if that's being driven by CCR1. And again, can you turn that off, that chemotaxis of the metastatic cells, can you turn that off with CCR1 inhibitors? So that's usually what I'm doing my research on for CCR1. With FFA2, I'm actually working with an investigator who's at UIC, who's an endocrinologist. And so he's interested in FFA2 and its role in diabetes. So I've been helping his lab screen compounds for that are biased for their role in being able to be useful in a type 2 diabetes model.
Wow.
So,
And yeah, I was listening to you talking about, chemotaxis and CCR1 and all of these, and then I remembered reading a paper about the CCXCR4, and I think that was your paper at that point. And that was the first driving how it was driving the relationship between CXCR4 and SDF1 or CXL12, driving cancer cells out of the primary tumor going into secondary tumor sites. And that caught, I think that was the first ever scientific paper I had read, and I loved that paper. It's although it's a devastating disease, and as you mentioned, a lot of people die out of these secondary tumors at distant sites. It's still a beautiful pathophysiological event that happens, and it's a smart way of getting cells out of one place to taking them into another. So you mentioned that you do screens. What kind of screens do you do? What kind of tools do you use on an everyday basis to try and answer some of these questions with these two receptors, for example?
So it's pretty interesting because even though we've certainly not coordinated this in any way, shape, or form, that several things have come up in your prior people who you've interviewed, right? And so one of them that came up had to do with native systems, right? The importance of using primary cells and understanding the signaling that happens in the endogenous receptor versus in a hex cell where you overexpress it. So I've been pretty much saying that for at least 15 years. Like I have a paper about it 15 years ago because I think it's absolutely true, right? So I try to take an assay that's no different than what's used at Big Pharma, right? They're doing calcium assays, cyclic AMP, right? These are common in my lab. And the difference is that I do it as close to the native cell as I can get. So I'm not using stem cell differentiation, though I'd love to be able to do that. But I'm at least using a cell line that's relevant to the disease that I'm looking at. So if it's multiple myeloma, I'm using a multiple myeloma cell line. I'm not using HEC that overexpresses CCR1.
Yeah.
If it's diabetes, I'm using a pancreatic cell line. I'm not using a HEC cell with FFA2 expressed. So I try as best I can to get to something that's closer to a native system when I do my screens. But I don't think otherwise that I'm doing anything that's different than what other like companies would be doing.
Mm-hmm.
But I do look, just because of my background, I look early on for bias.
Okay.
So GQ versus GI versus beta-arrestin, and I look at that relatively early to find out whether that's going to play an important role in the physiology and the pathophysiology of the disease itself.
Mm-hmm.
So, you know, I might not want to turn off GI coupling for FFA2. I only want to turn on GQ.
And
So,
and are these, for example, in the context of FFA2, is this this need for bias, is it known that it's important to have if you want to correct the situation, or is it just something that you want to document to better understand how the receptor works?
yeah, it's not usually known. Sometimes it is, right? Sometimes they know. So in the case of FFA2, we pick GQ because they know that calcium is important for insulin release, right? So it has nothing to do with the actual, and they know that FFA2 signals to GQ.
Mm-hmm.
So when I got to sort of pick my bias, I'm going to pick something that's going to turn on GQ. And it was only sort of, that, you know, like we say that in the grants, so I'm looking for something that's GQ biased. But it turned out that it looks like that FFA2 is actually constitutively coupled to beta-arrestin. So I think we found that if you have an inverse agonist of FFA2 beta-arrestin constitutive activity,
Mm-hmm.
You actually then push the receptor down the GQ coupling. So I don't have to have a GQ agonist. I can have a beta-arrestin inverse agonist and get the same result.
Totally. But then here's my question when it comes to this bias. And, you know, bias was, and it's funny because, as you mentioned, I think the discussion about HEK cells versus more native-like cells was with Terry. And then, yeah, I think even with Paul, we brought up this question of bias, and he was mentioning that he just came back from a talk of Laura Vaughan. And it wasn't planned, but when you're looking at bias in general, I mean, there was for quite some time now this idea that you wanted compounds that would, you know, direct to G protein versus not beta-arrestin to yield a physiological response that you want without side effects or less side effects. So what are your thoughts on that, knowing that right now the recent publications indicate that it's not as black and white. It's more that you want to fine tune the response. I just want to know, what are your thoughts on it?
I don't even know that it's not black and white. I think the problem is that you're not going to find a compound that's going to give you black and white, right? So while you can turn off GQ coupling by having a knockout system where you just knock out GQ, right? That's not usually something that you can do in a person, right?
Yep.
You can tell that I can't do that, right, in a person. So the question becomes, how much is enough in terms of turning it down? How much is enough, right? Is a 20% reduction of beta-arrestin signaling enough? Is a 40% reduction in GI signaling enough? And I think it is probably cell dependent. So much of and this was mentioned, the allosteric modulators are often of lower affinity. There's often sort of a higher background because they're going to interact with more than one GPCR. And it's really sort of understanding what does that mean in the context of the whole system, yeah. Right? Because in every cell type, it's going to be different in terms of what interaction it's having based on the conformation of the receptor in that particular cell, depending on how many other receptors there are, depending on, you know, what kind of other proteins are present, right? Does it have other proteins that cause it to dimerize? Does it have other proteins that change its signaling, right? I mean, so one of the big things that I talk about, I used to give a talk about the native system and why it's important, right? A lot of people, for example, study neuronal signaling. But they don't do that in neuronal cell lines. They do it in like a hex cell line, which may not have GO, may not have GZ. These are known to be important in neuronal signaling. So you're like, you're not even looking in the right system, right? I mean, how can you know what your ligand, what your compound is doing to the ligand receptor interaction if you don't even have the right proteins present intracellularly that would normally be there?
Agreed. And then you mentioned, to your point, you mentioned that in order to look at the importance, for example, of GQ signaling with the FFA2 or any other GQ potentially receptor, you would just knock out that GQ, which you can do in a human person, in an organism or larger organism. But the problem is that cells adapt, and you knock out one G protein, the cell is going to find another way of activating that same pathway, or at least mimicking it with another G protein. So knocking out GI1, alpha I1, will definitely help the cell use the other two GIs
Right.
in order to do what it wants
Or
to do.
GOs.
I'm sorry?
Right. Or GOs.
Oh, yes. I think they're very plastic in that sense, and that shows how even the human body is very plastic in that sense, where you have a disease and you kind of change and you adapt. Your body adapts to that disease state, which brings me to your point where you were mentioning that you're looking at a specific GPCR function in cell lines that are closer to that disease state that you're studying.
Right.
And I think we can all agree on the fact that having someone who has a diabetes for 10 years definitely changes their whole body, their whole GPCR signaling, and their whole system. And studying cells that have a similar composition in receptors and in effectors as well might give you a better idea on how to target or how to screen for better therapeutics.
Right.
Which gets me to my next question. As we know, we all have genetic variations of different receptors or different effectors. How do you think those genetic variants will influence GPCR therapeutics?
So I think that they probably have influenced GPCR therapeutics just unknowingly, right? Unwittingly. Just like with the biased GPCR for the beta-2 adrenergic receptor that was found, cardiovol, that was more impressive in terms of how it treated disease. And the only thing, one of the things that they identified was the fact that it was biased, right, for beta-arrestin versus G protein signaling. That they didn't screen for that. It was just fortuitous that it was found to be the most effective when used therapeutically. And later on they found, right, that in fact it was biased. But I think the same thing may happen with drugs that target GPCRs that are already used, right? So there's a phenomenal paper by Hauser that came out just a couple of years ago that talked a lot about pharmacogenomics of GPCRs, and one of the receptors that they specifically looked at was the mu opioid receptor. And so they made what are known variant mutations in mu, and then looked to see what happens for drugs that are available now, right, that are already on the market. So a certain percentage of people already have these mutations that are, they are taking these drugs, there's no question. And so then, you know, some of the patients will respond, some of them won't respond, some of them will respond better, right? And so I think that that it's already out there in terms of having drugs that in fact may be more relevant based on your pharmacogenomic background. Mm-hmm. But I think more needs to be done. There's no question. There are so few papers out there where a company who already has a drug, who knows what the target is, where the target is a GPCR, where they've gone in and looked at what are the common mutations that are identified in the population at large, and what happens with our drug, with that receptor in those populations. It's just not done. Yeah, but it should be. It absolutely should be.
That would help also better understand why would one person react better to a drug versus another? And also we would allow to adjust doses or maybe adjust the formulation or even use a totally different compound.
Yeah, absolutely.
Based on your genetic, GPCR mutation or expression
Right.
in the body. And the other thought that I have about this is, for example,
I'm going to interrupt you, and I'm going to tell you, I think one of the most interesting things, right, in that Hauser paper, they talked about which of the GPCRs have the most mutations. And almost all of them have to do with like hormones, right? So FSH, LH, right? So for all the people who are having infertility issues, right? Is it as simple as that they have receptors where there are mutations where their body doesn't respond as well, for example, even to endogenous hormones, right? And so they ovulate differently because of that. I mean, it's just fascinating to me that this is already out there and known, and yet there's very little information if you do a PubMed search.
Yeah, and to your point, thanks for interrupting, because that's where I was kind of going, is not only better understanding the differences from person to person when it comes into their mutations in their GPCRs at large, but also how does the environment influence that?
Yeah.
What you eat, what you drink, or how much you exercise. Can you do something about it as a person? Sometimes it's not possible, but I firmly believe that trying to kind of figure out the best of all of the worlds, what you eat, how much you exercise, what you do on a regular basis, definitely can help, make drugs, for example, more bioavailable. Drugs, you would need less drugs if you have a faster metabolism that you can control through exercise, through food. And I think that's also something that needs to be studied. I think there are not enough studies about the effect of a drug on a specific patient with a specific genetic panel or mutations in their GPCRs, for example, and compare that to their lifestyle.
Yeah, because I think that things like diet, right? I mean, they know that GPCRs are influenced by the cholesterol level in your membrane, So certainly having a different cholesterol amount in your diet is going to alter the amount of cholesterol in your membrane. You're going to, like, how does that alter your GPCR in terms of its stiffness and how it can, in fact, bind to the ligand or not bind to the ligand, signal to the G protein or not signal to the G protein. So I think that environment is certainly part of the issue.
Yes, but it's hard to control. I was at some point talking to a doctor about this, and I was told that it's very difficult to control because you would first of all, building this kind of studies is difficult, but also you would also rely on what the patient tells you, the participant tells you, which is,
you don't have to do it in patients, right? You can start in mouse models, right, where you get to feed them whatever you want.
Yes,
right?
yes, and it's funny because I was having a conversation with one of my future guests who will end up releasing the podcast pretty soon, John Stryker from University of Arizona, and he had a student who decided that he wanted to study. So they study opioid receptors and pain, and they wanted to see what happens if you intermittently fast the mice. And how they react to pain, and can you give them less medication, painkillers, if they're in a fasting state. So, and the answer is yes. They didn't change their food, they didn't change their diet, just got them on intermittent fasting, and they recorded a significantly reduced amount of drug needed to treat the same amount of pain inflicted.
Which is amazing.
It is because I did ask, so did you give them something else to eat? And they said no.
Right, it's the same diet, just different timing.
Exactly. And can you think about the effects if it would be, different timing, but also a different diet, and comparing all of those.
Right. High fat versus not. Yeah.
Yeah.
No, I think that, I think it's one of the coolest things about GPCRs is how much we still don't know, right?
Yeah.
How much things we still don't know. So as much and as wonderful as it's been to get crystal structures and be able to use modeling capacity and molecular dynamics, that's fantastic. But there's still so many questions that we have no idea what the answer is. So, yeah, it's one of, I guess it's one of the best things, right, about GPCRs. There's always something to look at.
Yes, there is. There is. And speaking of crystal structures and tools, so we've spoken about using more native-like cells and getting crystal structures to better understand how the receptors work. What do you think are some tools or initiatives that could help speed up targeting GPCRs in drug discovery, but also understand GPCR biology at large?
So I'm a big fan of like the BRET technologies and the biosensors where you're looking at protein-protein interactions. And I would say that the reason I'm pretty fond of those has to do with dimerization. So Terry Hebert was on, right? I think that his work with dimerization and that understanding of receptor dimerization, it really, we still don't know though, physiologically, does it make a difference, right? If you have a ligand, does it bind better to the dimer, the homodimer, the heterodimer? That's amazing to me that still so little is really known about the physiological role of dimerization of GPCRs.
Yeah.
So I think all of the tools that are being used to explore that, you know, hands down, phenomenal. They've really helped us understand, you know, why, what, you know, doesn't make a difference. And they still have a long way to go, but at least we're trying. We're sort of scratching the surface, if you will, for the information that could be available to us.
Absolutely, absolutely, and I think it's a tough question to answer. We already know that for some GPCRs, like the GABA B receptors, you need that dimer to function and to get up there at the plasma membrane. But what are the implications, and how can we leverage what we know or what we can know about their function in treating a disease setting? That would be that's definitely something that we absolutely need to do. That gets me to my next question. And I put this question in because I'm always interested in not the simple one-word answer, but I'm interested in more getting a perspective. So the question is, do you think GPCRs are still good drug targets? And I think the answer is yes.
Yes, and I think they will be for a really long time. I don't know that we're ever going to run out, right? And what's interesting, I think, actually now is that several of the orphan GPCRs, right, so GPCRs that they don't know what the endogenous ligand is, they now have compounds that target them, and those compounds are being used to treat diseases. So independent of knowing if or maybe there's not an endogenous ligand, although that seems amazing to me that there wouldn't be, that you're able to target a receptor, not even knowing what turns it on naturally in the system, and yet you can still use it for disease. I mean, that's amazing, right? And so I think that many of the pharma companies have sort of seen that, recognized the value of that, and are currently working on sort of their own orphan GPCRs in the background. So even if we don't know what the disease is, I say there's a big interest in antibodies for GPCRs, which is pretty interesting, right? There are several diseases that are caused by antibodies to GPCR, right? Graves' disease, for example. But the idea that you could use an antibody for a GPCR as a therapeutic is pretty new. It's pretty novel. But we know, right? I mean, I come from an industry background. We know that you're going to be much more likely to bring an antibody to market than you are to bring a small molecule to market in terms of success, right? When you start down the pipeline, it's much more likely to be successful if you have an antibody. And with that in mind, you know that the targets are good for the GPCRs. You know that they're physiologically relevant. So why wouldn't you want to target them with a monoclonal antibody? So I think that is really an area that's ripe for being exploited by drug companies. I think dimerization is still of interest, but they're sort of on the fence as to knowing whether they need to target them or not.
Yeah.
I think that there's still quite a bit of interest in GPCRs. I think there's currently, right, and has been for a while, interest in things like allosterics, biased modulators, inverse agonists. Those all are still being looked at, being explored. for disease states, and I think that they will be continued to be of interest. I think that pharmacogenomics isn't explored enough, but I think I'm not in tune with what all the industry players are doing right now, but if they're not already on that bandwagon, I suspect that some of them will get there.
Definitely. Thank you for that. Yeah, I think it's there's so much to do. And speaking of orphan receptors, I mean, I'm not even sure that we've discovered all of them yet. So I think there is much more to be discovered. I think last month or the month before, there was a new orphan GPCR that was recently cloned. And when you think about cloning GPCRs, you think, 1990, early 2000s, when you had that huge wave of cloning basically anything. And I think it was Silvio who was mentioning that at that time, whenever you cloned something and you looked at its function, you automatically or very easily could publish it in a high impact journal like a Nature or something. We're not there anymore, but being able to document more GPCRs, more orphan GPCRs, but then again, understand which are their endogenous ligands and what they do has definitely a potential of increasing GPCR-related drug discovery.
Yeah, for sure.
Super. So you went from wanting to go to medicine, doing a master's, working at Pfizer, then working for a company, and then going back to academia. You're well seasoned in both worlds. What would be your advice to junior scientists who want to contribute in the field but don't really know where to start?
I think I tell people all the time, I think that if you're a scientist, you have to follow your passion. Science is nothing that you get into because you want to earn a lot of money. To me, being a scientist is like being an artist. You do it because you can't imagine your life doing anything else. I do science because I love science. I love asking a question, right? I get to find an answer that nobody else knows, potentially. I get to make new DNA molecules that nobody's ever looked at before. I mean, it's amazing to me that I get to do this for a living. So I tell students all the time, don't do science because you're going to think it's going to make you money. You have to do science because you just love it. And so my advice, follow your passion, whatever it is, whether it's GPCRs, whether it's tyrosine kinases, whether it's cancer signaling, you have to do what you have interest in because the hours are long and the pay is bad. And so as long as you like what you're doing, that's okay. I still go home happy every day. It doesn't matter whether the experiment worked. I mean, I'm always in a better mood if it does, but I'm still happy that I got to do the experiment. So.
And you get to ask the question that you're interested in at that moment.
Right.
We've been talking science and GPCRs for quite some time now. Are there any moments in your career where you did an experiment or you went to a talk and then you realized something that you could call an aha moment as a scientist during your trajectory?
Yeah, so I guess I have a couple. So I would say the first one was when I was in Heidi's lab, and I read a paper about the bark C-terminals tails, and they had actually taken out the bark C-terminus and they put it into a plasmid and they made these tools. And it was after reading those that I decided I wanted to do that with the G proteins, and I made the mini genes that many people use in their labs. And at the time when I read the paper, I was like, I can't believe this hasn't been done before, right? I mean, it seems pretty simple. They did it with 55 amino acids. Why can't I do it with 11? Not realizing, of course, that 11 amino acids was actually much harder because then you can't find it because it's so small. You don't know if you have it expressed or not. Yeah. But at the time, I thought it would be really simple. And so I started down that course. But that was definitely an aha moment, why don't I try it kind of thing. When I first did a screen when we were looking for the high affinity C-terminal peptides in Heidi's lab, we would use those peptides when we looked for small molecule screens. And the first time that we did it, I was looking for antagonists. I was just looking for something that blocked the high affinity peptide from binding and then would block the G protein from binding, right? That's all I thought about. But what I actually found were agonists, things that made the G protein bind better. And then the question is, what do those do to ligand binding, right? So some of them make the ligand binding better, but they don't signal because the G protein is literally stuck to the receptor, right? So that was definitely an aha moment. I had no idea that I would find agonists, biased agonist as well as antagonists the first time I did my screen. And so I think it's really important, right, when you have a finding that is completely unexpected, that you follow it, right? That you ask the question, why did that happen, right? Why did that happen? And I think that's important, instead of being like, oh, let's try it again, maybe there's something in there, right? So I think that's pretty important. I would say the third one for me would be the recent one with FFA2, when we found an inverse agonist for beta-arrestin, because those had never been reported before. And so they've been reported for a mutant vasopressin receptor, but not for normal receptors to have endogenous beta-arrestin activity. So I had to call around and maybe have you ever heard of this? Do you have this in your lab? I mean, I don't know what to make out of it. And so again, when you get something that you can't really explain, it's really to say, okay, why? Why did that happen? Let's follow that. And I have to say that academics really let you do that, right? I can ask that question. So.
Yeah, agreed. And it's funny because whenever you get a result that you didn't expect, the first thought that I have in mind is, did I do something wrong? How come? And then you end up repeating it and you get the same results again, and then that's when the thinking actually hits in, and you can go a different route and potentially discover something new.
Right.
Which is so phenomenal that you get to do the experiment, you get to discover, you get to doubt yourself at the moment when you get the result. Did I copy and paste this properly? Did I pipette properly? And then be like, no, actually this is exactly what's happening in the system. All right, I have two last questions for you before we close our current podcast. So one of them is, it's a topic that comes up lately in the news. And what are your thoughts about increasing diversity in the field?
So I think it's a tough question, right? If it were an easy question, we would have had an answer a long time ago. When I was a postdoc a million years ago in Heidi's lab, I used to volunteer for a program that was called Wild About Science. It was part of the American Heart Association. And I would go to inner city schools and give them a demo on science. And I remember being amazed at how excited they got. And so I think that anything that we can do to stimulate just that intellectual curiosity about why did that happen, I think that you're going to inspire many kids. It doesn't matter where they're from, right? It doesn't matter what their background is. And so you're going to have more diversity the more that you target younger children and you tell them it's okay to just ask the question, why did it do that? Or what would happen if? And you have to really ask those sort of open-ended questions and let them explore.
I like that answer. And I think, increasing diversity in general starts at a very young age, when you give kids that opportunity to experience new things and to see new things, and to also get the ability to ask those questions. Because if you've never been exposed to science, the chances that you're going to get interested in science are smaller than if you have, both parents, professors, doctors, or whatever. Not that I'm saying that it's good or bad. I'm saying that having that opportunity is also very, very useful and very important to everyone. One other thought I had about diversity and is mentorship. So it's very important to be a good mentor, but also to seek mentorship, because long term it also builds confidence. You're not born 100% confident. I think you learn to be confident, and having that mentor and that support system is also very important in order for anyone to go step into that next thing. Doing a master's and then deciding, oh no, I want to do an MBA, or I don't want to be a doctor, I want to work at Pfizer, for example. That's also very important.
I think that's absolutely true. I think it's just even having somebody to talk with, to bounce ideas off. So when I was at Northwestern, Paula Stern often served as my mentor and did for years after I left Northwestern. And whenever I had a scientific question, I would she was my encyclopedia, right? We all have those people that I didn't want to go look it up in PubMed. I would just call Paula and be like, so I read this paper and I don't understand why. And she always had an answer for me. Or she still does. I mean, what am I talking about? She still does. I can talk to her about anything. And so that was really helpful to me, because so much of what you do in science is like in the minute. Like, I want to know that, right? We're sort of instant gratification kind of people, right? It's really hard for us when we have to wait overnight for cell growth.
Blame dopamine. Blame dopamine there.
So I'm like, I really want to know right now, right? I don't want to look it up. I don't want to wait an hour. I don't want to, so it's so nice to be able to call somebody and like talk science, right? I think we live in a relatively small world. I don't have a lot of outside friends that are scientists, right? People ask me what I do, my kids, right? The other parents, nobody else is a scientist. So I think that really mentoring, having somebody to talk to, having somebody that you can ask their advice, it's a phenomenal thing to have. So
yeah, it is, and even in having that trustful relationship where that even if you feel like your question is silly or is a stupid question, you can still ask that person, and that person will give you their honest, direct opinion, and you won't take it in a bad way. You actually will learn from it.
Right. No judgment. There's no such thing as a stupid question. Yeah. No judgment.
I don't think so either. Thanks, Annette, for your time. I wanted also to ask you, when you have job openings in your group, where do you advertise those? And before you answer that question, I just wanted to let you know and let the audience know that we also have a career page, drgpcr.com/career. And the intent behind that page is to kind of serve as a yellow pages for all potential GPCR related jobs, because they're hard to find when you're out there.
So I work for Midwestern University, and Midwestern University has a website where they post all their jobs, and I would say people go there. I'm also on Twitter, as are you, right? And I'm GPCR scientist, and I will often retweet job openings of other GPCR people, because I think that, right, we live in a pretty small world, and anything I can do to promote that is a good thing. So I think that's another place, right? Follow Twitter. You or me, you're going to always have careers up. So I think that's helpful as well. And I think that sometimes people just have to do the initiative and like literally write the person they're interested in. So Paul said that on the week that he did it, just write me. And I think that's absolutely true, right? If you have a passion in that area, then you should find out who's doing the work and you should write them. Because even if they don't have a job now, if they get a grant, then they're going to think of you, right? So they may not even need to post the job per se. They can call you, you can talk about it, right? I mean, you really have to go to people completely blind and say, I'm interested. Whatever you have, I'm interested.
I think that's great advice, and that reminds me of how I met Paul. I think it was about 5 years ago, and I wanted to do a second postdoc in his lab. And he had said, well, I'm not sure if I'm going to get funding. You can come out. And actually, I was introduced to Paul through someone else and the name escapes me, but it will come back in a second. And I decided to just, go to his lab and ask, go there, ask him ahead of time, can I come in? I really want to, meet your team. And I gave a talk, and he ended up not getting the grant that year. I never went to Paul's lab, but then we kept in touch.
Right.
And that relationship started out of me taking the initiative of sending the email and then going there and meeting him, meeting his team. And, he kindly accepted to be my first test subject on the podcast. And I really, really very much enjoy talking to him. And that's how kind of the whole podcast idea, started out from that email that I sent over 5 years ago saying, hey, I love your work. Can I come and visit your lab?
That's exactly right. I mean, and you don't even know where the relationship will go in the end, right? You have no idea of knowing in 10 years what your research is going to be and if you're going to be crossing paths again and again and again, right? And it's because you form that relationship that you're able to call them and be like, oh, hey, I got this problem, right? I found this weird finding.
Yes, and you never know. So to the junior scientists out there, just, send that email, read the paper, pick up the phone. It's a small GPCR world, and everyone is always welcoming. Send a nice note saying, hey, I read your paper. I really liked it. Do you have 5 minutes?
Right.
And who doesn't have 5 minutes to talk about GPCRs?
It's true. And if they don't have 5 minutes, that's probably not the person you want to talk to.
Yes, that's the other side of the coin. I mean, you can't get a positive answer from anyone, and that's not only, a GPCR crowd thing, it's everywhere. And that's okay too, because then it stirs you to a different direction that you wouldn't have taken if you wouldn't have sent that first email from whom you didn't get an answer. So just don't be afraid to go out there.
No, definitely not.
All right. Thank you, Annette, for your time. I really enjoyed our conversation. Take care and be safe.
Thank you. It was a pleasure.
Thank you. Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Subscribe to the podcast wherever you listen to podcasts. I'd like to thank our guest, Attila Forrest, Jin Chong, and Shivani Sachdev. Music by Rosa Bershish. I'm your host, Dr. Yamina Bershish. We're always excited to hear from you. Visit us at drgpcr.com or send us a note at hello@drgpcr.com. I also wanted to wish you happy holidays and a very happy new year. I'd like to thank this year's guests and thank you for being such a wonderful audience. Thank you again for the privilege of your time, and until next time, stay safe.